Okay, so let's start this session. I'm really happy having with us this morning Christine Tardif. Christine Tardif is assistant professor at McGill University. She is in biomedical engineering and neurology and neurosurgery department at McGill since 2017, I think. She is also a co-director of the MRI unit at the McConnell Brain Imaging Center, and she is also a co-director of the Quebec bioimaging networks. So she is very interested and developed a research program around how we can characterize the tissue within the brain, and those characteristics involve a new technique in MRI, high resolution and quantitative MRI to do this kind of very important research in our field. She has particular interest in healthy subjects, but also neurological diseases. She worked on how we do this quantification of myelin in subject during the lifespan. So it's a very, very important point to analyze the structures, but also the physiology of the brain. So she has also a lot of extension of method regarding high field MRI, like 7 Tesla MRI in animal brain, but also 3 Tesla and 7 Tesla MRI in human. So Christine, thank you very much for coming today and giving us this lecture on contrast mechanism and quantitative imaging. Thank you.
Thank you. Thank you very much, Abid, for the invitation to be here and participate in this 2-week school, which is quite unique. Very nice initiative that you guys have put together for the community and for the students. Alors, je réalise qu'on est à l'Université de Montréal, au Centre de recherche mathématique. Je suis francophone. Je vais faire ma présentation en anglais. Si vous avez des questions en français, ça me fait plaisir d'y répondre en français. If you didn't understand that, that's fine. I was just saying that I'm happy to take questions in French for those who are from l'Université de Montréal, because I speak both languages. Okay. So as Abid said, I'm from across the hill, so at McGill University. And my research lab really focuses on the development of MRI techniques. And this spans quite a few phases of the imaging process. So from the development of contrast mechanisms, fast acquisition techniques and things like that. So really image acquisition. Then we work a bit as well on the image reconstruction phase. So how to go from the Fourier domain to an image. Once we have the images, we work on some biophysical modeling in order to get some, microstructural parameters of the tissue, and then image analysis as well. And really the main driving force of my research is my interest in myelin plasticity, because we know that it's not only neurons that are plastic, but the glial cells play a huge role in plasticity as well, including the oligodendrocytes that form myelin. And in studying how this myelination may become maladaptive in different neurological disorders, including neurodevelopmental disorders. So that's kind of my research interests. Of course, we won't be talking about all of that today. So I'm going to start today with more of an educational seminar. Starting at the basics, okay? So I realize that not everyone here has a background in MRI. So we're going to talk, go over like a crash course of what MRI is, and in particular, what quantitative MRI is, and how we can start to look at the microstructure features of the brain using quantitative MRI, okay? And then after a well-deserved coffee break, we're going to tie that back to the general theme of these 2 weeks, which is the neural networks, okay? So how can we use these techniques to start to characterize more comprehensively the neural networks that everyone are studying and modeling their function, okay? All right, so there aren't very many of us this morning. So, you know, and I love that because I generally teach grad students, not undergrad students. So I'm used to smaller cohorts in the classroom, which is really fun. So please feel free to interrupt me at any time, okay? I can clarify concepts. I don't have an hour and a half of material this morning. I have more like an hour. So the point is that we can stop and go on tangents if you have any interests or things that you'd like to clarify, okay? All right. Okay. Right, so we're going to start at the beginning, talk a little bit about conventional MRI, okay? So these are the type of MR images that you would acquire in the clinic. So of course, there are so many scanners, you know, across the world and in the province, bit of a lack of scanners actually in Quebec and a long waiting line, so that's another story. And these, because MRI is, you know, non-invasive and widely used for diagnosis, for monitoring disease progression, as well as for monitoring the impact of treatments, right? And so here are a few examples. Of course, what I love about MRI is the fact that it's such a versatile imaging modality, right? You can acquire many different types of images with it. So here we have two different examples. The first one is multiple sclerosis. So this is a, you know, neuroinflammatory and neurodegenerative disease where you'll see plaques that start to accumulate in the brain. And MRI is actually part of the diagnostic workup to, you know, for multiple sclerosis, and has been, I think, since at least 2010 officially. And so you can start to see these lesions here in the brain that accumulate, right? And so that's what they'll do in the clinic. And what we started doing in research is quantifying this disease that we see in the brain, right? So what can we do to quantify it? Well, how many lesions are there? Simplest thing, right? The radiologist can scroll through the 3D volume and count the number of lesions. A little bit more advanced, what is the total lesion volume? So for that, we need to start to segment the lesions, right? And quantify the volume of each in comparison to the total volume of the brain, right? How does that volume or number of lesions progress over time? So those are the types of metrics that we'll maybe look at in a clinical trial to see if a new treatment is working in this subject, right? Is a lesion growing or shrinking? So these are quantitative metrics, right, to characterize a disease, but they're still extracted from conventional weighted, well, I'll refer to as weighted images that we get in the clinic, okay? So this is often what we see. In other cases, you know, we're not really looking at these types of features, we're looking more at patterns. So these are leukodystrophies, more like genetic diseases, okay? And we're looking at different patterns that we see in the brain, and that's used to kind of try to distinguish what the disorder is, what's causing it. Okay, so this just brings me to the field of MRI-based computational anatomy, right? So for those who've done MR imaging, and you maybe are familiar with this, but this is kind of like the bread and butter of structural MRI research, okay? It's really widely used across the world. So the most common, you know, image that you'll get is a T1 weighted image. And this has been harmonized across all the scanner manufacturers, you know, the different field strengths. So it's really, you know, like a robust image contrast that a lot of different groups use. From that image, a lot of different image analysis tools have been developed in order to segment it into its different compartments, right? So here is just a very simple, you know, tissue segmentation where we're trying to extract the gray matter shown in green, and that's overlaid on the original image. From that, you can start to extract different metrics, the volume of the different subcortical gray matter structures, for example. But you can also look at the cortical thickness, which is now for the same subject, just projected onto a 3D reconstruction of the cortical surface, right? So now that you have these tools, we can start to look at more subtle differences between people, right? So instead of looking at a lesion where we can really do this at the individual level, we can start to look at more subtle systematic differences that occur during normal development, during aging, during learning, and things of the sort. So here are a few, you know, seminal studies. So there's one here by Gagte that was just looking at how the volume of gray matter changes during aging, right? And so it actually decreases as you go from about 5 to 20 years old. And this is part of the normal, you know, neurodevelopmental maturation process. So this is wonderful because it's, you know, captured at the population level, right? And now we can see, for example, in the context of disease, Alzheimer's disease, we know that we have cortical atrophy in this neurodegenerative disorder, but it's hard sometimes to see at the individual level. But using these quantitative techniques that are really robust, really repeatable, we can start to say, well, that maybe in an AD cohort, you have accelerated, cortical atrophy in comparison to neurotypical subjects controls, right? So these are really subtle effects that we're starting to pick up with quantitative techniques. And one of my favorites, of course, is this learning-induced plasticity. So this was done by Bogdan Dragansky's group, where they basically taught a cohort to juggle for 3 weeks. And they scanned them before, after the 3-week juggling, and then they did a retention scan a few weeks or months later. And they were able to see local changes in gray matter volume that were induced by this training, right? And this was compared, of course, to a control group that did not undergo the juggling training. So this is just to show you, how powerful these quantitative techniques are. Of course, all the examples that I gave you so far were all done in the context of research labs. What I think is a really exciting direction is this, direction done that's been taken by Richard Bethlehem and, of course, a whole group of colleagues, which is to create brain charts. So for those of you who have kids or remember yourself going to your pediatrician when you were young, right? There's a growth chart. Are you meeting, the expectations in height and weight for your age and sex, right? Are you following that curve during your development? Well, now there's like an initiative to pool data from across the world using these computational techniques, right? To start to get what are considered normal, normative neurodevelopmental brain charts. And we can start to look at the individual subjects and see how they compare to these charts, and perhaps even identify people who are at, risk for neurodevelopmental disorders or other things because they're kind of falling off their trajectory, okay? So I guess I wanted to start with this because I wanted to acknowledge the fact that there are a lot of quantitative MRI tools out there, and metrics that are derived from clinical images, and they're great, and they've had a huge impact on research, and soon will have an even greater impact in the clinic. But when I'm talking about quantitative MRI, that's not really what I need, okay? So here are some of the limitations of the techniques that I talked about. The main limitation is interpretation, right? You have a change in apparent, MRI-derived gray matter volume. What does that mean? What's happening at the microstructural, level? How do you interpret that? Well, it's really hard to say. There's a lot of different things. Just in the context, this is a really great review article by Robert Zatori. So just in the context of normal, healthy plasticity, a lot of things can be driving changes in gray matter volume, right? Whether it's axon sprouting, dendritic branching, neurogenesis, but even glial changes and angiogenesis can cause a change in gray matter volume in your brain. So the idea here is that we want to develop MRI techniques that are more specific to the microstructure and composition of the tissue, mainly so that we can improve our interpretability, right, of these images. Okay, so here are kind of the 3 motivations for this work. So as I said, the first one is to understand what the underpinnings of these macroscopic effects that we're capturing with conventional MRI are, right? The second big motivation is, can I detect changes earlier? By the time you have atrophy in your cortex, there's you can maybe intervene less, it's maybe a little late. You won't have as much of a disease modifying effect, right, with treatments. So these microstructural MRI may be able to detect these changes earlier before you have macroscopic changes that are detected with normal clinical MRI, and hopefully intervene earlier in the lives of these people, right? And then, of course, since you're working earlier on in the disease, the chances are that these quantitative MRI techniques will have a greater correlation with changes in behavior and disability. Okay, so these are the general motivations that I have for working on more quantitative MRI methods. Okay, so this is what we're going to talk about for the rest of this first lecture. Point 2 is really your MRI crash course. Okay, we're not going to go over everything about an MRI. We're not going to go over spatial encoding, things like that. Just really where the signal comes from. What are the contrast mechanisms? What do they mean? Okay. Then we're going to go into how to go from a conventional weighted image to a quantitative map. And we're going to start to relate those maps to the microstructural features of brain tissue. I'm going to finish off with some more recent research that starts to look at multimodal biophysical models to really, take a step further and improve the interpretability of our images. All right, so back to basics. I think you need to understand just, there's just a few slides on this so that you really have an appreciation of where the signal comes from, okay? So what are we imaging in MRI? Well, we're looking at nuclei. They have either an odd number of protons and or an odd number of neutrons, because these nuclei will have a property that's called spin, okay? So it basically means, as it suggests, that they're spinning on themselves, right? Around that long axis. And once they start spinning on themselves, you'll basically create a circulating electric current, right? And that will help you generate a little magnetic field, okay? So we refer to this as the magnetic dipole moment. And so this, spinning nuclei will end up behaving effectively like a mini magnet, right? With a north and a south pole, okay? And basically, the strength of this magnetic moment is related to the gyromagnetic ratio, okay, which is a feature of the nuclei. So basically, if it has the faster it's spinning, the stronger the magnetic moment, and that will be also determined by your gyromagnetic ratio. Okay, so now that you have these mini magnets that are everywhere in your body, right? What happens when you enter a magnetic field? Well, as soon as you enter like a static magnetic field, which we'll refer to here as B0, which will be the main magnetic field of your scanner, right? Your mini magnets will basically start to, align with that magnetic field, and there'll be like a force, so a torque that's applied to these mini magnets, and they will start to precess. So you'll see here that it's, it continues to spin about itself. In addition to this spinning, it'll start to precess around the main orientation of that external magnetic field. And these are actually in different directions, okay? And so this frequency of precession, which we'll call the Larmor frequency, is actually proportional to the gyromagnetic ratio in gamma there, times the magnetic field, right? So this is why in the field of NMR, you'll sometimes hear about field strengths, especially when you talk about scanners. But if you're talking about like, benchtop spectrometers, they're often, referred to in terms of megahertz, right? So it's because you're actually referring to them as with regards to the Larmor frequency of the system as opposed to the field strength. Okay. So, of course, the gyromagnetic ratio will be different for the different nuclei that are in the body, okay? And we know that the frequency here will be determined by the total magnetic field, right? And so why do we need to know what this Larmor frequency is? It's kind of a hard question, but maybe in case some of you guys worked in MR. Because this is, you need to be working at this frequency to start to interact with these mini magnets, okay? With these spins that are in your body. So if you want to excite the spins, you need to be exciting them at this Larmor frequency. When you acquire the signal, it'll be at the Larmor frequency, so you need to demodulate it. So you really need to know the frequency of the spins that you're interested in. And of course, it'll vary with field strength. So if you're working at 3T, the Larmor frequency of your spins will be lower. So you're typically at 128 megahertz for hydrogen spins at 3T. And it'll be, as you change the field strength, it'll be higher or lower. Okay. So you have these mini spins that accumulate in the body, right? And they're all randomly oriented when you're outside of the scanner, okay? Now we're going to start to play with them a little more and see where we get the macroscopic signal from. So as soon as you put them into the main magnetic field, they're all going to align approximately with or against the main magnetic field, okay? And basically what happens is that, without going into too much of the, physics behind this, they can be in two different energy states, okay? So this is quantum physics. So you can be in the low energy state where you're basically aligned with the main magnetic field, or you can be in the high energy state where you're aligned, opposite to the main magnetic field. Okay. And so when we're looking at when we're measuring the magnetization in the head, for example, what we're looking at is the sum of all of these little vectors, right? And so you need that sum will be basically proportional to the difference in population of spins that are in the low and high energy state. Okay. Okay. Now, what affects these this net magnetization? Well, of course, the bigger the energy difference between the states, the bigger the population difference you're going to have. And there's a couple of other things that will affect it as well. So, for example, if you want to transfer a spin from a low energy to a high energy state, right, so it becomes anti-parallel to the main magnetic field, that's going to require some thermal energy. So the more thermal energy that you have in your system, the more spins that will be in that high energy state. Okay, so what affects the equilibrium magnetization? So first of all, the equilibrium magnetization, which we're going to write as M naught here, that's the maximal signal that you can get, right, when you enter the scanner and you're looking at the magnetization, okay? That's the maximum you can get, and then we're going to start to manipulate that. And that will be, of course, a function of the main magnetic field, which is why there's such a rush and so much investment in working at higher magnetic fields and bigger magnets, right? It'll be proportional, of course, to the density of spins in the body. So that's one of the reasons why we're mainly imaging hydrogen, right? Because there's just so much water in your body. We can also look at other nuclei like sodium, phosphorus, and there is a lot of imaging, especially in the context of stroke, for example, for sodium. But the density of sodium spins is so low in your body that the image quality is much lower, right? This SNR is much lower. Okay, and it'll also be a function, of course, of your dry magnetic ratio and a few other parameters such as temperature. Okay. So now we have this, net magnetization, and we're going to start to play with it a little more by adding additional magnetic fields, all right? So the first magnetic field is the main magnetic field B naught. Now we're going to apply a second magnetic field, okay, which is 2 orders of magnitude smaller, so usually on micro-Tesla range. We're on the Tesla range here, typically, right? And B1 will be on the micro-Tesla range. And so this one is applied perpendicular to the first magnetic field, okay? And it'll actually be applied at the Larmor frequency. And this is your way of putting energy into the system, okay? So you're doing that, and you basically bring those spins from the parallel to the anti-parallel state, and you end up at the macroscopic view, okay, classical physics, flipping your magnetization vector, which is along the z-axis of the scanner, into the transverse plane, okay? Now, all of the signal that we're measuring in MR is in the transverse plane. Okay, your z-axis is like your memory axis. The signal's there, it'll recover there, it's great. It's kind of like a storage space, but you're not measuring it. If you want to measure a signal, it has to be in the transverse plane. So once it's in your transverse plane, it won't stay there static, right? It'll want to return to its equilibrium position. Okay, so there's going to be 2 mechanisms that occur there. You're going to get recovery along Z, and you're going to get a decay in the XY plane, and that's actually governed by 2 different rates. Okay, so we're going to look at that. Okay, so here's just a quick animation. So in blue, you have individual spins, and then the Z and XY component. So we're almost at the beginning. Okay, it flipped down, and now you see the spins dephasing. You see the Z component in red recovering, and the XY component in yellow that decayed, right? And I think you notice that the yellow component decayed faster than the red component recovered. Okay, and that's always the case. And the individual spins are there in blue. So the other 2 are basically the sum, right, of all those little vectors.
Sorry.
Mm-hmm.
There is a big question that
all those spins, they are identical. So if they have the same initial relaxation, how come that they don't behave the same in terms of when you relax?
Excellent question. So it is a bit of a simplification in the sense that they actually aren't all perfectly aligned like that when they start
So you have
in general. Okay, so there is more of a distribution. However, they still dephase with respect to each other. That part of the animation is correct. Yeah, by interacting with the different spins. Yes. Okay, so I'll get to that. Yeah. Okay, so we'll first talk about T1 relaxation, then we'll get to this dephasing that he referred to. So how does this? So the T1, okay, is the constant that's used to describe the longitudinal relaxation, which is an exponential relaxation, okay? And basically what happens is that you need to think at the level of the individual spins here, okay? So they're basically flipping back from the high energy to the low energy state, okay? And so the realignment of these spins is happening through exchange of energy between the spins and the surrounding lattice and their environment, okay? And this is happening due to random molecular collisions, okay? And so this is what happens along the z-axis. So now let's say you're in the clinic and you want to acquire an image that has a T1 contrast. What you're going to do, right, is, here you have, after an inversion pulse, right, you'll have the recovery of the Z component and the magnetization. And here we have 3 different tissue types: white matter, gray matter, CSF. And so you'll basically choose a time where you have the maximal contrast, that you're interested in. And at that time, you're going to acquire the signal, right? You're going to put it in the transverse plane first, and then you're going to acquire the signal. And that's how you play with your T1 contrast and get the image that you want. Okay. Now let's talk about those spins. So in the transverse plane, we'll get T2 or T2 star relaxation, which is also referred to as spin-spin relaxation. And what happens is that basically these spins will interact with each other, right? And while they're doing that, they'll experience small deviations in the magnetic field, which are temporary, okay? And so that's what's going to cause, as soon as the magnetic field varies a bit, the alarm or frequency, the frequency of precession of the spins varies a bit, which means that they'll start to accumulate differences in phase, right? And that's why they start to spread out as shown in that animation. And because the magnetization is the net vector sum, is that these spins start to spread out with respect to each other, and that's why you get a decrease in the net XY magnetization. Does that make sense? Yeah. Okay, so that's what you have here. There are 2 different types of dephasing. So there's the spin-spin interactions, which will cause your T2, okay, decay. In addition to that, you can also have small variations in your magnetic field, which are like static and therefore reproducible, okay? So this can be simply because of, a spatially varying magnetic field, just because you don't get a perfectly stable magnetic field in the scanner. It can be because of, the composition of your tissue, like iron deposition, which will cause, static but locally variable magnetic fields. And so what happens is that this part is non-reversible, but this part is. And so we'll see how that works. Okay, so before we get into that, again, similar concept here. You're flipping the magnetization to the transverse plane, and your signal will decay exponentially, but at different rates, right? And so depending on the tissue type, and so you again look for the time at which you have optimal contrast and you acquire the signal. And there's a bit of a trade-off here, right? Which is kind of obvious based on the graph. You acquire your signal as early as possible, you get the highest SNR, right? So the best image quality, but maybe very poor contrast between your tissues. So sometimes there's a bit of a trade-off. Sometimes we wait and acquire the data at a longer, later during the decay to get the ideal contrast between our tissue types, knowing that we're losing SNR while we do that. Okay. Okay, so here's just an example of a T2 star weighted image that's acquired at 2 different echo times. The echo time is the time at which you acquire the data. So as you can see, the signal here is obviously much brighter because you're earlier in that decay process, but the contrast between gray and white is flatter, right? So here we've waited a little longer. Some of these structures have a shorter T2 star time, and therefore the signal decays much faster, and therefore they'll appear darker in this image, right? And this is mainly a few deep gray matter structures that have this feature because they're rich in iron. okay? And so then the contrast between the structures is much higher. Okay. So as I said, part of this is decay is reversible, right? So the images I just showed you are from a gradient echo acquisition. So it's really the simplest sequence you can have. So this is my 90-degree pulse. So just flipping your magnetization to the transverse plane, T2 star decay, and you acquire the data. This is the special one. It's called a spin echo sequence. It's like the second big category of sequences. And you add a 180-degree pulse. So the spins that we're kind of dephasing out like this flip and start to rephase back towards each other, okay? And this allows you to remove that second component, right, which is reversible, and end up with a purely T2 weighted image contrast or signal decay. So the best analogy for this is the running analogy. Imagine you have a bunch of runners and they're all, some are better than others, some run fast, some run slow, and you have them at the starting line. You blow the whistle, they all start running. After 30 seconds, they're all spread out, right? If at the 30-second mark, they all stop, turn around, and start running back, they should all arrive at the starting time block at roughly the same time, right? So that's basically what you're doing here. Okay, so here's a visualization, 90-degree pulse. They start to dephase in the transverse plane. 180-degree pulse, flip them around, and they rephase back and form an echo. And it's usually when they all rephase and form that echo that you want to acquire the signal. Okay? Okay. So similar concept, this is to give you an idea of what it looks like. Of course, here, after the 180-degree pulse, you're starting to rephase the spins, so you regain some of that signal, but you don't go all the way back to what your original magnetization was, right? Because you still have that non-reversible T2 decay. Okay, so you'll end up with overall a higher signal intensity, and you'll also avoid some artifacts which are caused by field variations. So again, if you choose a long echo time, you'll have enhanced contrast between your tissues. If you choose a very short echo time, you're closer to that equilibrium magnetization, right? And so you have less contrast. And basically the main contrast of your image will be the proton density, right? So how many spins, how much hydrogen do you actually have, right, in your tissue? Okay. So this is just, a bit of a recap, but also to show you the versatility of MRI. These three images, very different contexts, but were all acquired using the same and simplest MR sequence there is, which is the gradient echo sequence. Okay. This was acquired, this is actually a group average, which was acquired many years ago at the MNI. It's the MNI Atlas, acquired a 1.5T, it's T1 weighted gradient echo. This actually I acquired during my PhD. So a few years ago, post-mortem at 3T, okay? Proton density weighted, same thing, gradient echo sequence, just many, many hours. And this one was acquired a bit more recently on our 7T scanner, T2 star weighted. Again, so 3 different field strengths, same basic sequence, 3 different contrasts. So just highlighting, the versatility of the technique and how if you start playing with the acquisition parameters, you can really generate different types of images, right? So the only things that are varied here are your echo times, the time at which you acquire your image, which will determine the amount of T2 contrast or T2 star contrast you have. Your TR, that's the repetition time. It just determines how long do you give the system to recover back to its equilibrium position. If you don't let it return all the way back, well, then you'll have T1 contrast. And the flip angle plays with the T1 contrast as well. Okay, so that's your basic MRI crash course. Yes.
Question about the difference between the contrast. Which one's better? I mean, the application of the contrast or contrast.
I'm not sure I understood your question.
If we choose, for example, high TV, have high contrast, right?
Yeah. So you'll have high, yes.
And what's the difference between two different images?
Okay.
The application.
Yeah. So it really depends on what you want to look at, right? So in some cases, what we want to do is we want to, segment the cortex, right? So this is kind of a, that is a T1 weighted image, but here we're going to here. So this is a T1 weighted image. It's what's most commonly used to segment gray matter and white matter, right? So divide the brain into its tissue types, because it has just the clearest contrast, especially at the cortical boundary. So for that, you basically want to minimize the other sources of contrast. You want to make it T1 weighted. So you'll choose, a flip angle and a TR to optimize that contrast, but then you'll choose a short TE. You don't need T2 star contrast. It may even go against your T1 contrast, so you want to have as high a signal as possible. So you'll choose a very short TE in that case. In this case, the deep gray matter structures actually don't pop out very well. So if, you're interested in a disorder where those structures will be affected, like Parkinson's disease, you may decide, okay, forget T1. I'm going to go for T2 star because I know that these structures really have a nice T2 star contrast. So I'm going to, minimize my T1 contrast and focus on my T2 star, use a longer echo time. So it's really, it depends on the application and what you want to do with the image. So I guess MRI is versatile. You can acquire many different types of images, but I guess the other way of looking at that is that there's no one size fits all. There's no one image that you can use, to answer all of your questions, right? Okay. All right, so any other questions before we continue? No? Okay. So what do I mean by quantitative MRI? Okay, now the concept here is quantitative MRI, right, to look at brain tissue microstructure. So we're really quantifying the magnetic resonance parameters of the tissue, which describe its local physico-chemical environment, okay? So for example, instead of having a T1 weighted image, I want to measure what the T1 is of the tissue, okay? So a T1 weighted image, the signal intensity will be a very complex function, right? It's a function of your T1, of course, that's the main driver of contrast, but T2 star and proton density, which are other MR parameters of your tissue, will also contribute. Maybe less, but they're still there. Then all of your acquisition parameters contribute as well, right? Your signal will depend on the field strength you're working at, the echo time, the TR, the flip angle, all those things. Okay. In addition to this, we also have all your system imperfections, which complicate things. Okay. So now, whether I acquire, let's say I acquire an image at, Saint-Justin Hospital, and I acquire then an image at the Montreal Neurological Institute, they may both have different protocols for T1 weighted imaging. You can't directly compare those images and those signal intensities, right? Because there'll be different functions of all these parameters. So that's why instead what we're trying to do is get a quantitative map of what T1 is directly, for example. So that now becomes, a quantitative value that you can directly compare across different voxels in your brain. They can compare how it changes over time. They can compare to other subjects, right? So this is what I mean by quantitative MRI, right? But of course, in addition to what I showed before, there's also the methodological biases, which quantitative MRI is supposed to solve. They don't always solve them. Some quantitative MRI techniques are more robust than others, but generally that's the concept, right? And I'll just give you an example here that's very, one of the biggest problems that we have, which is the non-uniformity of the magnetic fields. So when I say, hey, let's apply a 90-degree pulse to flip our magnetization entirely into the transverse plane. Well, in reality, everywhere where it's 1 here, I'm applying a 90-degree pulse. Everywhere where it's 0.8, well, I'm applying 0.8 times 90 degrees, right? Because we're not able to achieve a RF excitation field that's perfectly uniform across the brain. And that problem actually gets worse as you start working at higher and higher field strengths, okay? And it'll end up not only in images, here I'm showing them in spectral view so that you kind of highlights the effect. Not only does the signal intensity vary throughout your brain, right? But the contrast between neighboring tissues will vary as well. And so there's been a lot of development that's been done on the image processing side, right? So that you can kind of apply a multiplicative filter, which will make this image more uniform to the eye, right? But the contrast here at the motor cortex, for example, where the field is lower, it's still lost. You can't recover the contrast that way, okay? And so these are the types of things that we get around using quantitative MRI. Okay, so of course, we discuss all the advantages of it, but there are some limitations. First of all, in order to do quantitative MRI, typically your signal to noise ratio efficiency is lower, okay? And that's simply because you'll have to acquire several images and fit it to a signal equation, right? So it just increases your total scan time to get a single map at the end, right? So it's less efficient. We're maybe willing to live with that in the research setting, but in the clinic, this is a really significant limiting factor. The second issue is that these techniques are not necessarily available as products by the scanner manufacturers, okay? So when I started my PhD in like 2006, we had to develop all these protocols and all these sequences ourselves. We'd have to often have master research agreements with the vendors in order to change their code in order to do this. The field has evolved tremendously. So a lot of the most of the techniques that I'm going to show in the rest of the presentation can all be run using product sequences on most scanners. Okay, so it's really changed over the years. And a lot of the software that you need to use to reconstruct the quantitative maps from the images are now available as well. Actually, at Polytechnique, there's kind of a champion of this, Nicolas Stikov, who's very into, open science from an MR acquisition and analysis perspective. Okay, so now we're going to dive into how we quantify these different features and what their relationships are with brain tissue properties. So it's not an exhaustive list. These are kind of the main mapping techniques that you'll see in the literature and that will hopefully be included more and more in open, public data sets. Okay, so the first one is the first example I gave, T1, okay? How do we do T1 relaxometry? There's a lot of different fast techniques out there. I'm just kind of going over the gold standard techniques today, just to give you an idea of how it works, okay? So here we're going to be using the inversion recovery technique. So you basically apply an inversion pulse that will generate your greatest T1 contrast between your tissues. And then after an inversion time, you apply flip angle to bring your magnetization to the transverse plane where you can actually measure it, okay? Now you repeat this several times, every time using a different inversion time. So for every image, you'll get a different T1 weighted contrast, right? So you're basically sampling this recovery curve. You know what your signal equations are, right? And they're a function of two parameters of your tissue. It's T1 relaxation time, and it's equilibrium magnetization. So two unknowns, you need, at the very least, two measurements along this curve. And then ideally more, and then you can reconstruct both a T1 and an M0 map, okay? So here are just examples of what these maps look like. And they're quite similar. That's the first thing I want to get across, right? They're really very similar. And that's because actually your T1, so your proton density map, this is the easiest one to understand and is actually the easiest one to interpret. It's really basically saying, how many hydrogen spins do I have in my tissue, right? So what is the water content of my tissue? That's what you're looking at, okay? Especially the echotimes we're working with. Whereas T1, here, if you recall, it's really the interaction of the spins with their surrounding lattices, right? So T1 will be highly dependent on water content, on the amount of spins that are there, and then on the macromolecular content of the tissue, okay? All of the bound spins. So there is a linear relationship between your T1 and your proton density. So here you have 1 over T1, and here 1 over your water volume fraction. And you'll see that it's, of course, very linear, and this is over a whole group of subjects. This is work by Aviv Mezer from Jerusalem University. Now, the catch with proton density, of course, is that you're not really measuring proton density, you're measuring the equilibrium magnetization. So we need to calibrate it. So in order to do that, typically what you'll say is you'll assume that CSF is 100% water, and you'll simply calibrate your measurements to your CSF, okay? And then if you simply do 1 minus proton density, you'll end up with your macromolecular tissue volume fraction, okay, which is more and more used in literature. One point that I want to, which will come up later, so just keep it in mind, is that we're looking at MR visible protons, okay? So that's why I said hydrogen spins, we're looking at the water content. But actually, there are some hydrogen protons that are bound to macromolecules that we can't see directly using MR, but they're there. So that's why we talk about MR visible, okay? All right. Okay, so now for the next steps, I just need to, before we talk about, T1, yep.
So I was looking for the relaxometry in general because you are hitting potential.
Mm-hmm.
So you say you need minimum of 2, but in practice, how many are you using? Are you using 2? Are you using more? Because the
Good question.
The accuracy of the fit will be enhanced. And I assume also that to do the fit, you linearize the
Mm-hmm.
Just to see where is the sensitivity or what kind of range of inversion recovery you should use to get an accurate.
Yeah, it's a good question. So again, I'll mention just 2 techniques that are really widely used that are efficient. Of course, efficiency is critical in the clinic. So if we can get away with 2, we'll get away with 2. So the first one is a variable flip angle, right? So it's a grain echo technique with a short TR. And so you can vary the TR, but you're mainly varying the flip angle to vary your T1 contrast. It's the same concept. We typically do 2 measurements, 2 flip angles. The other one would be MP2 rage, which you may be familiar with. It's slowly starting to replace the MP rage sequence as an anatomical scan, and you can get a T1 map from it as well. 2 inversion times, 2 measurements. So I think that this is really, I think that it's appropriate if you're looking at the healthy brain, or if you're looking at a brain where you know that the features will change a lot, but the range, of T1 times for white matter and gray matter is quite restrained. So the idea is you optimize your flip angles or your inversion times for that range of T1s that you're interested in. CSF has a really long T1. So any voxels that have partial voluming effects with CSF, you may get a bias or an error there, right? What happens if you're looking at disease with edema? Boom, your T1's gonna off the charts. So now the choice of inversion times or flip angles may no longer be appropriate for specific lesions. Some people use T1 mapping to monitor gadolinium, right, uptake. So, well, again, you're looking at quite a change in T1 when you give a bolus of gadolinium in the brain, that enters the brain. And so maybe your technique isn't appropriate for that, So it's really about, I think, knowing what is the range of T1s that you're interested in, and then targeting your acquisition parameters to that. Yeah. But if you don't, so just an example where things go wrong really often is postmortem imaging. So you have beautiful protocols to do relaxometry in vivo, and then they have a postmortem brain, and they want to scan it really high resolution to validate. The relaxation times of the brain shorten drastically, postmortem and with fixation. All your protocols are off. You won't get the right measurements at all. So that's the case where you need to, typically what we'll do is we'll do a really low resolution scan with many flip angles or many inversion times, get a gross estimate of what the range is we're looking for, and then do high res with 1 or 2 measurements that are at the right, targeting the right range. Mm. Right. So, okay, so I'm bringing, this image up now because, we need to know what we're looking at in the brain, right? So here what you have is basically an electron microscopy image, right, of a cross section of a myelinated axon. And I'm bringing this up because you need to know what the environment is, right? Myelin has a huge impact on MRI contrast, on all contrast mechanisms, okay? So the relaxation times will vary depending on where you are, right? So whether you're within your axon, in the extracellular environment, or if you're in the water that's stuck between all of these layers of the myelin sheaths, your relaxation properties are going to vary, right? So, and that's because these sheaths are formed of macromolecules, of proteins and lipids that the spins can interact with a lot, okay? And so this is going to affect all of our contrast mechanisms. And so T1 maps are, I'm going to say this because it's not said enough, are not specific to myelin, but they're very sensitive to it. And so if you're looking in a healthy brain, you'll see some beautiful contrast that really highlights the myeloarchitecture of the cortex. So this is a high resolution T1 map acquired at 7T. It's 0.5 millimeters isotropic. And already you see, the strong contrast between gray and white matter, obviously, because the white matter is really rich in myelin, and that affects your T1 time, it shortens it. But there's some hidden contrast in the cortex there. So a trained eye would be like, oh my God, the cortex here is so much darker than all the way up here. But it's hard to see when you're looking at your volumetric image. So you can take this data and project it onto a cortical surface, which we then blow up here, okay? So you can see the data that's hidden deep in the gyri. And you'll see it's the same data. And you'll see that, the primary areas, visual, motor, auditory, all have much lower T1 times because they're more densely myelinated than the higher association areas, okay? And this kind of nicely replicates the classic myeloarchitectic work that was done by the Vokes and their disciples, and that was kind of put together really nicely by Dr. Newen-Hughes more recently into the MNI space so that we can start to look at that with neuroimaging. But again, you're capturing a metric that is sensitive to myelin, and so you see the myeloarchitecture features of the cortex, but you're not only sensitive to myelin. As we saw in the previous slide, you're also really sensitive simply to the water content of your tissue. So here's another, before we're looking at data from 1910 and comparing it to MRI maps that we've acquired recently. This is nice, work that was done by Nicholas Bach while he was at the NIH. With Alonso Silva in the marmoset. So the marmoset is a very small New World primate, roughly the size of a squirrel, okay? And what's nice is it has a nice smooth cortex, less gyrification, so it's easier to look at. But it really has similar myeloarchitecture to the macaque and to the human, right? So you'll see these areas, first of all, the bands of Baillargeau, which is there are two bands of dense myelination within the cortex, right? And you'll see some primary areas that are densely myelinated. This is area MT or V5. It's extrastriate visual cortex that's responsible for processing like rapid motion in your visual periphery. Densely myelinated in humans, but really densely myelinated in non-human primates. And you can see that in the myelin stains and in the in vivo MR maps that were acquired in the same animal, okay? So I think this data is online where you can really look at both data sets simultaneously. Okay, but again, just can't drive home enough that T1 will be affected by water content, inflammation, and actually, as we'll see a little later, you see here, a third aspect of tissue microstructure that will affect your T1 contrast is iron. Okay, and so this is beautiful work done in Leipzig by Carsten Stuber, who was part of Bob Turner's group, where they took a postmortem brain section of the motor cortex and scanned it high resolution, got T1 and T2 star maps and everything. And then they did some elemental mapping, so of iron and of myelin. And what they do here is that they assume that we're in the context of fast exchange. Okay, so you're like in a little homogeneous environment. Okay, so they do make that assumption, but then your relaxation rate, which is just 1/T1, is a function of the relaxivity of water, and then the relaxivity of iron times the concentration of iron, and the relaxivity, of the macromolecules, mainly myelin, times that concentration. And so if they use that model, they're able to really simulate the MR maps that we actually measure using these elemental maps of iron and myelin, okay? And are able to really show that, I think, over 70% of the contrast within the cortex in this context is driven by the variation in myelin content within that area. So this is for the cortex, okay? They did not extend that analysis to the white matter. Okay, so one thing I am going to point out, that M here is, I say macromolecules, I think they say myelin, but it's not only the myelin that's contributing, right? We know that there is myelin in the cortex. A lot of people forget that, but there's about 10% of the myelin that there is in the white matter, right? There's a lot of other cellular membranes with proteins and lipids that can be contributing as well. Okay, so similar concept, you know, relaxometry. Here we're looking at different echo times. The advantage, of course, is that this is a much more efficient acquisition because you don't have to acquire a whole image, you know, do a whole acquisition for a certain T1 contrast and repeat it. After a single flip angle, you can acquire the signal several times to get your different echo times. So the whole, you know, T2 star mapping process is much more efficient than T1 mapping. If you want to do this for T2 mapping, you have to, of course, include several inversion, refocusing pulses, right? To always refocus that dephasing back. But it's the similar concept. Okay, so this is the example I gave earlier. So again, this is 7T data acquired in Leipzig when I was there. We're looking here at a T1 weighted image, and this is a T2 star weighted image. So we're not the quantification yet, but you really see the enhanced contrast here in the globus pallidus, for example. And in the next slide, you see, of course, you know, substantia nigra, red nucleus, all these structures that are really rich in iron, where all of a sudden, you know, it's very easy to segment them from the neighboring tissue, right? Now, similar concepts can be applied here. So your R2 star relaxivity, again, similar linear equation, right? Relaxivity of water, contribution of iron, contribution of myelin. What's interesting here is, of course, is that, you know, myelin is diamagnetic and your iron will be paramagnetic. And so they will both reduce your R2 star. They'll both shorten your T2 star, increase your R2 star. But they have actually different magnetic properties, and we'll get back to that in a minute. So here, this is beautiful work by Fukunaga, postmortem again, where we have a myelin stain, an iron stain, and then our T2 star map, okay? And you really see similar features across them, right? Your band of Baillet, you know, more dense iron in the white matter, especially near the border with the cortical gray, right? So this iron distribution really varies depending on where you are in the brain. And basically in the cortex, the reason why, you know, the iron map, T2 star map, and myelin map are all really similar is because in the healthy brain, most of that iron is actually in the cell bodies of the oligodendrocytes that are forming the myelin sheath, right? So they're effectively kind of co-localized. So yes, if you're looking just a healthy brain, if you did a T2 star map of the cortex, you'd effectively get something that looks like the myelin architecture of the cortex. But that's not what people use T2 star mapping for. They typically use it to look at iron content. So if you're looking at aging or a neurodegenerative disease where there's an accumulation of iron in the tissue, you'd be looking at not, you know, the patterns across the cortex that will still look like the myelin, but you'll be looking at differences, you know, within an area across subjects or over time. And that could give you an idea about the accumulation of iron in that structure, right?
Yeah. How come that you don't see the iron in the blood vessel of the hemoglobin? It's maybe less dense.
It may be how the tissue is prepared, because it's more, it's all postmortem. It's all postmortem. Yeah. So I'm assuming that it's just, yeah, washed away. Yeah. Okay. All right. And this is actually just showing that, you know, on the cortex as well, those same primary areas stand out. This is actually work by one of our local researchers, Jean Quenadal. Okay. All right, so back to this concept that we have a heterogeneous, environment, right? So all of these equations like this make the assumption that we are in a fast exchanging environment, okay? So in the next slide, we're going to talk about slow exchange. The idea here is that on the time scale that we're doing our measurements, okay, the hydrogen protons, the water that we're actually imaging, is not going to go from in the cell to in the myelin sheath to out the cell, okay? So those compartments are like, exchange between those compartments is slow. So they're kind of like different sources of signal, right, that can be summed up within a voxel. So that's why you have this sum here of the signals from the different compartments, okay? And so we can use this, and instead of doing, assuming that we have a monopolar exponential decay in our signal, because we have a single T2 or T2 star species, we are now going to say, okay, well, these different environments could have different T2 relaxation times, and they do. And we're now going to do a densely sampled, acquisition, and, using invert the Laplace equation here, and we'll end up, oh, sorry, guys. Not sure what's happening there. Do you not see the next slide? Oh. I think I have an issue with the Zoom. I'm going to try it again. Hold on. How did I get to the Zoom? I think it just died on me. I'm going to just try to join up.
Oh, it's because you don't have internet anymore.
But I had it. EduRoam should work.
Maybe just disappear. Otherwise, you can connect to demo cliff. Oh, yeah, maybe there is a problem with EduRoam. You can connect with the other one, Udemy with cryptage, if it doesn't work.
Oh, this seems to work.
It came back.
No. Let's see if I connect. Yes. I'm going to mute. I'm going to share screen. And this. Ah, yes, we are back. Okay. I was wondering why we were not timed in terms of slides. Okay, so the idea here is we're going to do, forget the idea of just sampling 2 points. You can't do that. Here we're going to do a very dense sampling of the signal decay, right? And then not assume that it's a monopolar exponential decay. We're going to inverse the Laplace equation, and we're going to look at a distribution of the T2 star or T2 star times in the voxel, okay? And we know from a few experiments that were done that the T2s of the intra and extracellular water are actually very similar, okay? And the T2s of the myelin water is much shorter because you're stuck between those sheaths, and so there's much more spin-spin interactions, okay? And so you can then simply look at this distribution and assign a T2 to each compartment, right? They're going to say everything that's below this cutoff. This is a simulation, so it's very clean. The real data is not this clean. But you're going to assume, choose a cutoff and be like, okay, everything below this is my myelin water. Everything above this is my intra and extracellular water. And then you can just compute the area under those curves. And you end up with a myelin water fraction, right? So that's kind of an index for how much is myelin and how much myelin there is in your voxel, for example. Okay. So in this case, you're still doing a T2 relaxometry experiment, but you need to acquire a heck of a lot more data. Okay. And this technique was actually pioneered by Canadian Alex McKay, who's in British Columbia. So this is one of my favorite studies by Cornelia Lowley, also in British Columbia, where they compared that to a postmortem sample of tissue from a multiple sclerosis patient. So here really where you have a and looks all fast blue, which is a myelin stain, okay? So this is your MR image, your stain, And you really have just a beautiful agreement between the data, spanning not only from, normal appearing white matter, diffuse appearing white matter. So that would be like the kind of like dirty white matter that you see here, where there's diffuse pathology, right? Your lesions and your gray matter. And it's just really beautiful work. But again, this technique is really challenging to do in vivo because it just requires such a long scan time. Okay, so far I've been talking about relaxometry techniques. We have 15 minutes left, where we've been looking at the water, okay? As I said before, there's hydrogen protons that are bound to macromolecules, right? But we haven't been talking about those much because basically what happens is that their T2 relaxation time is so short, on the order of microseconds, that we can't image them using MR, okay? I didn't go into spatial encoding, but basically what happens in MR is that you excite the signal, then you have to turn on some gradients to localize where the signal is coming from in the volume, and then you acquire the data. Okay, so your echo time's on the order of milliseconds. The T2 time is on the order of microseconds. That signal has gone, okay? So you can't image them directly. So what we're gonna do is that we're gonna get at them indirectly, okay? So we have this two-pool model, right? So we have the free water pool that we're already imaging, and then we'll have this semi-solid pool, or the bound pool, okay? These are the hydrogen protons bound to these macromolecules, okay? And we're gonna make them interact with each other. So what we do is we apply a magnetization transfer pulse, an off-resonance pulse, so not at the resonance frequency of the hydrogen protons that are in water. We're going to apply it off resonance, so 1, 2, 3, 4, 5 kilohertz off resonance, and this will actually only affect these bound macromolecule hydrogen protons that are bound to macromolecules. So they will become saturated, right? And then what's going to happen is that they're going to interact with this pool that we can actually image. And so the magnetization will be transferred, right? This is our equilibrium magnetization of our free water pool, will be transferred from this pool to the saturated macromolecules. And so you'll get a decrease in the magnetization of the pool that we can actually see. Okay, so we apply this pulse as like a prep pulse, okay, to like prepare the system, and then we acquire the data. Okay, so this is an indirect way for us to look at those bound macromolecules that have a really short relaxation time. Okay, so there's different ways of doing this. The simplest way is magnetization transfer ratio. So you acquire an image without and then with this MT pulse, okay? And then you just look at the ratio of this, okay? And this is what your map will look like. I don't think this projector really does it justice. It looks very quantized, but that's what it looks like. And if you do a much more complete two-pool model, it's a long, long acquisition time, but you can actually get what the bound pool fraction is. And you see here that there's a decent correlation between your MTR and your gold standard bound pool fraction. And this is because it's a bit of an oversimplification, okay? We know that the T1 contrast is actually counteracting our MT effect. And we'll also have, as I said before, variations in your B1 field, right? Which will have an effect too. So by acquiring 1 or 2 more scans, we can actually measure the MT saturation, okay? You're correcting for these 2 confounding factors. All of a sudden, your contrast between white and gray is much more enhanced, and your correlation with the bound pool fraction is much stronger as well, okay? So this is what I would say is most commonly used in the research field because it's a relatively efficient scan, and it really is a good index of what your bound pool fraction is, okay? Now, in the interest of time, I'm going to skip over some of these things. So this is really one of the more, I say the community would accept the MT-SAT is a more specific marker of myelin, but it's not entirely specific. There's better techniques, okay? But it's definitely a step better than any of the relaxometry techniques. And that's because the myelin effect, right, of course, the MT effect is caused by the macromolecules. And in the myelin sheath, you have roughly 70% lipids, 30% proteins. And in the other cellular membranes, it's the opposite ratio, okay? And the lipids tend to cause a much stronger MT effect. So that's why when you're in the white matter, you know that your MT signal is really driven by the lipids that are forming this myelin sheath. When you're in the cortex, we have a lot less myelin. The interpretation, again, is not as clear, okay? Okay, so we looked at a lot of different, maps. It takes a long time to acquire all this data, but thankfully there's now protocols such as this multi-parametric mapping protocol, where basically you can acquire a few different images which can jointly be used to reconstruct many maps at once, okay? So this protocol, which is kind of coined by Nikolaus Weisskopf, but really builds on the work of Gunther Helms, when it was originally proposed, I mean, this is like 10 years ago, it took 20 minutes to get all of these maps 1 millimeter isotropic in the brain. We're now, using even more parallel imaging, compressed sensing, all these other techniques to bring that scan time down even further. So I think we're at the point where it starts to become, feasible to include these in large research studies and potentially even in clinical research, okay? And there's really a push to make that happen. Okay, this is just a slide that wants to highlight my word of caution, which I put out now in all my talks because I see a lot of people using, big data sets like the UK Biobank and the HCP and extracting all these measures and making really big claims about what they mean. I would say always report what you're analyzing and then discuss the interpretation, right? But for example, all of these MR contrasts, put them on the surface, they all look like the myeloarchitecture of the cortex, and anyone can claim, hey, I'm measuring myelin. But we know that that's not the case. And we know that, T1 weighted over T2 weighted signal contrast, for example, beautiful work by Matt Glasser and that whole HCP team, really helped to segment the cortex based on its myeloarchitecture. Absolutely. Would a change in this signal between a patient group and a control group mean that there's a change in myelin content? Not necessarily. Not necessarily, because it's just not specific enough. And that's true for all of these techniques, actually. So it's really important to understand what is the MR property you're measuring, what is contributing to that contrast mechanism, and how does that change in the context of the healthy brain and in the context of pathology, right? So you really always need to know a lot about the disease that you're studying in order to interpret your MR maps. Now, nevertheless, last step, only 2-3 slides here. I have 7 minutes. There have been efforts to kind of combine these techniques, right? Now that we can acquire several of them efficiently. So, of course, you can do this in a statistical way, and just say, oh, I have a higher MT here and a lower T1 there. What does that mean? But I'm talking about biophysical models where we really put all of this information together to improve, our interpretability further. So I'm just going to give you 3 examples. This one was really promising, but still needs a bit of work. So the idea here, you saw these images on the equations on the previous slides, simple linear equations, right? That say that the relaxivities and the magnetic susceptibility, okay, is a function of the water content of the tissue, myelin and iron content of the tissue multiplied by the relaxivities, right? So if we have these 3 measurements, 2 unknowns, we can basically now reconstruct maps of the myelin and iron content of the cortex, okay? So I love the idea. This work was done by José Marques. I'm not sure I pronounce his name right because he's Portuguese, but anyways. And I think the idea is really interesting, and I think they're continuing to follow up on this work. You'll notice that the maps don't really look like what you expect. And I think that's mainly because the myelin here is not actually the same in all 3 equations. In one case, it's more macromolecules, and in another case, it's really more the whole structure of the myelin sheath. So it's not really apples with apples. So this does need a little further work, but I think it's a really exciting direction that group and others are looking into. A second exciting direction, which was actually developed in Montreal, is looking at the G ratio. Okay, so what is the G ratio? The G ratio, like my beautiful sketch here of myelinated axons, It's the relative thickness of the myelin sheath, so relative to the caliber of the axon, right? So in terms of math, it's really just the ratio of the inner to outer radius of that myelin sheath, okay? And there's a reason why you want to measure this. It's because it, the myelin density in a voxel can remain constant, but it doesn't mean that the myelination of the axons is the same, right? So we have a nice case where the G ratio is different, right? You clearly see that the myelin sheath is much thinner, so the G ratio is higher because you have partially remyelinated axons, like in the case of MS. Same myelin content in both voxels. Here, there's no, demyelination, but there's axonal loss, right? So this would look the same in a myelin map, but in a G ratio map, it'll look different, and it can have a huge impact on, how you and how you interpret that finding, right? So in the context of MS, for example, it can tell you, okay, well, are there axons left that I can remyelinate, and how well is my remyelination therapy working, right? So really interesting in that context, but in other contexts too. So how do we measure this using MR? Well, first of all, we use a very simple geometric model. Okay, so we're not measuring the G ratio of individual axons, obviously, because of the resolution that we're working at with MR, right? We're getting an aggregate measure for the entire voxel. Okay, so that already has its limitations. Nevertheless, it's really just a simple, geometric model, assuming that they're cylinders. And the model becomes a function of the myelin volume fraction and the axonal volume fraction of your voxel. So then the challenge to the MR community is to measure those two variables. So we're not measuring the G ratio directly. We're measuring the axonal volume fraction, the myelin volume fraction, and then using those in that simple geometric model, okay? We talked about a lot of different techniques here to look at the myelin volume fraction, okay? There has been work that have used all of these, even proton density, to look at that. I would argue that T1 is not specific enough. If you do MTSAT or myelin water fraction, I think those would be more appropriate. But here you really need to make sure for this to work, for the G ratio to work, that you have, unbiased, accurate measurements of the myelin volume fraction. You can't just have something that's correlated with it. And then I did not talk about diffusion today because I know that Maxime Decouteau was here earlier this week, right? And probably gave a fantastic lecture on diffusion imaging. And so there are a variety of diffusion models that can be used to quantify what the axonal volume fraction is of a voxel. Now you combine these two and you can get your G ratio. So here's just an example again in the context of multiple sclerosis from their seminal paper, where you can see myelin loss in the 3 lesions that appear really nicely here in this flare image acquired in the clinic. You see a loss in the axonal volume fraction as well. This is what we used to do in MR. Now we're taking the time to put those together using the G ratio formulation. And what we see is that we have these 2 lesions actually have a normal G ratio, right? So the axons that are left probably well myelinated. This one, on the other hand, right, has a much higher G ratio, indicating that the axons that are there probably have a thinner myelin sheath. So that could be indicative of remyelination of some of these axons, because when you remyelinate them, typically the sheath is a little thinner. Okay, so an interesting direction. This is the kind of seminal work that they had, but there's a lot of course, given this promise, there's a lot of research groups that have looked into this further. So I encourage you guys to look at the publications on that. All right, and my very, very last example is diffusion relaxometry. So trying to get to the relaxation times of those different compartments. Already we looked at T2 relaxometry and getting the T2 of the myelin water and of the intra and extra cellular water, remember? So the issue there is that T2 of the intra and extracellular water is very similar, right? They overlap, so you can't, in the slide I had earlier here, see, this lobe is for the intra and extracellular water. And it's really like two loads, but they overlap so much that you can't really find the limit between the two, right? So using, you know, a T2 relaxometry experiment alone, you can't differentiate those two compartments within your voxel. But using diffusion, you can, right? Because the diffusion of water within the axon is very different from the diffusion of the water in the extracellular space. So what we do in diffusion relaxometry studies, right, is that we actually use this contrast and diffusion to help us measure what the T2 values are of those two different compartments, okay? So basically, in a nutshell, what you do is you repeat your diffusion experiment at different echo times, and then you fit that to a single equation. Obviously, it's very long. That's a challenge for the community, you know, to work on, to accelerate these techniques, but that's the general principle, right? And then what you get is you get a map here. This is by Mark Nielsen's group oh, I forget right now, in Lund, where you're looking at the stick fractions. So that would be the intracellular T2, and the Zeppelin fraction, so that would be the extracellular T2, okay? And you can see that the T2 here in the white matter is actually lower than the T2 of your stick fraction. And then another group did a very similar measurement, similar study. And again here, instead of really modeling it and trying to extract the signal from both compartments, they just said, we're going to work at a really high B value, and we're going to kill this compartment, okay? So all of this, you know, because of the diffusion. So most of their signal is coming from the intraaxonal compartment, okay? And again, they do that diffusion-weighted relaxometry approach. And here we're looking at the intraaxonal T2 times for different white matter tracks in the brain, and repeated here for different subjects. And you can see that there is contrast in the T2 intraaxonal time between these different tracks in the brain, which is really interesting, and it seems to be consistent across subjects. So what does that tell us about it? Is the T2 time related to that compartment? How? The size of the compartment, its shape? There's actually really interesting work going on in Derek Jones' group in that direction. Okay, I'm 2 minutes late. Quick summary, just to say that, you know, quantitative MRI tool is really useful to look at the microstructure and composition of your brain, but that there's no one-to-one mapping with the biological features that clinicians are most often interested in. So we really want to acquire, you know, ideally several parametric maps, and jointly they'll give you a more comprehensive picture of what's going on. And of course, using more advanced multimodal biophysical models, we can start to really try to isolate the sources, you know, of this signal and what's happening at the microstructural level. And that's it. And we'll happy to take questions, but otherwise maybe over coffee, and we can chat about the future directions that my lab is working in after that.
Okay, thank you very much.
Maybe you can start with one or two questions, then we can continue the question over the coffee. So
I lost my.
question from the audience.
About validations, and when you have your biophysical models, so at some point maybe you would like to validate what you measure. I mean, like trying to look in microscopic experiments if it's true or not. Did the community try to do that, or? Yeah,
that's a great question. The easy answer I would say is so first of all, we happen to be organizing a 3-day virtual workshop that starts next week, which is on QMRI validation. Okay, so in case anyone's interested in that, I'll post the information later on, or Abid can share it with the community. But yeah, so there's a lot of work on that. And I think it can't be, people need to come at it from different angles, right? So a lot of work can be done using sophisticated phantoms. So phantoms are like test objects, right? And there's some really nice work being done, for example, by Aviv Mezer from Jerusalem University. He's a chemist, I think, by training. And so he's creating some really cool phantoms, with lysosomes and everything to validate some of the MR techniques we're looking at, So I think that's really interesting in the context of like looking at cellular structures and membranes and things like this. If you want to just validate how accurate you are, at measuring T1, well, kind of the question that Christophe was getting at earlier, okay, you're shortening your protocol, you're still doing a good job. There's more simple phantoms, like NIST has a phantom where they have, a series of vials with different T1s and T2s, that you put in your scanner and you can check. But that's kind of a simple question. I think your real question is, in the context of pathology, does this hold the interpretation? So that's where I think these cool phantoms that Aviv is making are really helpful. I think that we need to do a lot of preclinical imaging effectively. So, in mice and rodents, in non-human primates, and validate them. And not only in healthy animals, but in disease models to try to validate our techniques, So it doesn't have to be the exact model you're looking at, but if you know that a huge confound is microglial activation and you want to know how that affects your MT, well, find a mouse model that has that. It doesn't need to be the same disorder, and look at how that confounds your measurement, So I think there's a lot of work that can be done there. My pet peeve is that some of the work I showed today on validation did post-mortem MRI, and all of these parameters changed drastically post-mortem and with fixation, and the relationship of them with the histological features of interest also changes post-mortem with fixation. So I really think that the imaging in the context of validation still needs to be done in vivo as much as possible. So one of the things we're doing, for example, is that we started working with marmosets, and whether it's to look at the white matter or the gray matter, they're just really fantastic animal model. And we've been imaging a marmoset longitudinally, with many different MR contrasts that we've accumulated in vivo. And now we're looking at the histology. And again, we can't do, just T1 mapping and just MBP post-mortem, right? I think on both sides, you have to do a realm of MR parameters, and also probably a few different histology techniques so that you don't only look at the feature of interest, but at all the confounds as well. So it's not easy work to do that validation, actually. It's kind of, students get really excited about it at the beginning because it's a very disciplinary project, but there's a lot of challenges associated with it. So, yeah.
Excellent. Another question? No, I have a very small question related to also to the validation process. So it's really a good idea looking at the diffusion productometry and trying to link it to the myelin or to the G ratio and to link it also to the myelin. But again, when we do that, we are looking at diffusion, and we would like to make a reasonable link with the myelin, and this comes with, of course, validation somehow and creating a model to do that. But the idea is, it's a good idea. So, what is your, belief that this kind of function, sort of relationship between myelin and the contrast between water molecule is the right way to do quantification for the myelin?
So what's my favorite contrast for the myelin? So I don't have a single answer. I think it depends. Something that I've been doing is I have kind of two approaches in my lab. One is really to look at the white matter, and one is to look at the gray matter, because I think the confounds are different. So in the white matters, especially in the context, which is what I'll talk about in the next seminar, in the context of neuronal networks, the major confound to me is crossing tracks. In a vox, in over 90% of voxels in the white matter, you have crossing tracks. So if you take a measure of the myelin, you don't know which tract you're imaging. They're both contributing. So it's like this massive blurring kernel. So that's why I think diffusion has a role to play there, to disentangle the properties of different tracks. In the cortex, I think the confound is different. It's not the same. It's no longer an issue of architecture and geometry. I think it's more about specificity. You have myelin in the cortex. It has a really important role. Its role is actually quite different from its role in the white matter, but there's a lot less of it. So you need to have a contrast mechanism that's even more specific, So we have different techniques. If I say myelin imaging, I'm like, well, I acquire two different maps, one that's for the gray matter, one that's for the white matter. They're different because I think that they just have different issues. Yeah.
Okay, thank you. So we have a few minutes for coffee break, and then we come back for the second part of the research lecture of